Welcome back to the OIS Podcast. Register today for our Dry Eye Innovation Showcase happening on March 11th that will feature 8 company presentations and 2 panels that will give you insight to both industry and clinical perspectives. Visit ois.net/events to reserve your spot. If you missed the live panel discussion from our Gene Therapy Innovation Showcase, you're in luck. We've got the 5 retina specialists from our meeting right here to get you up to speed on the current landscape of gene therapy and how the sector is evolving to extend beyond treating just inherited retinal diseases. Listen to hear about promising clinical trials and how our panelists foresee a future with effective and less invasive treatment deliveries for diseases like wet AMD, diabetic macular edema, and retinal vein occlusion. So much to learn. Let's get started.
I'm going to ask each of the panelists to introduce themselves. Maybe I'll start with Peter.
It's a pleasure to be here. Thanks for the invitation. I'm Peter Francis. I'm the Chief Scientific Officer and Therapeutic Area Head for Ophthalmology at 4D Molecular Therapeutics, which is based in the Bay Area in California.
Well, thank you, Peter. Jeff?
Yes, I agree. It's wonderful to be here, Tom, and thank you for the invitation. My name is Jeff Heier. I'm the Director of the Retina Service and Retina Research at Ophthalmic Consultants of Boston.
Thanks for being here, Jeff. Arshad?
Hi, Tom. Thanks for having me here too with this great group of experts. I'm Arshad Khanani. I'm Director of Clinical Research at CRI Associates and Clinical Associate Professor at University of Nevada, Reno.
Thank you. Jose?
I'm Jose-Alain Sahel. I'm an ophthalmologist specialized in retinal dystrophies. I'm currently the chair of ophthalmology at the University of Pittsburgh Medical Center and also the former director of Vision Institute in Paris.
Thank you for joining us. Glenn?
Hi, Tom. My name is Glenn Yiu. I'm at UC Davis. I'm an associate professor and a clinician scientist. Our research lab studies gene editing as well as gene therapy in age-related macular degeneration.
Thanks, Glenn. As many of you know, in retina, we are so extraordinarily fortunate to be the epicenter of so much gene therapy innovation. And most amazingly, to have had the first US FDA approval of any gene therapy for a genetic disease occur in our own space. There are several important evolving innovations in retinal gene therapy, including exploring non-gene specific approaches for hundreds of rare inherited retinal diseases, transferring genes that are too large for AAV vectors, producing proteins in mitochondrial disorders, knocking down and replacing genes in autosomal dominant disorders, minimizing ocular inflammatory response, and investigating novel methods of ocular administration. I'm going to ask each of the panel members some questions related to some of these important and exciting topics, and I invite everybody to join in. But why don't I start with Peter? Peter, so as you know, AAV, the most commonly used vector, has provided a great platform for subretinal gene therapy, but has limitations around cargo capacity and inflammation. Can you update us on some of the innovations in vector design to address these limitations?
Yes. Thank you for that question. As you say, AAV has become the workhorse for gene therapy for ocular disease treatment. And it has a number of very attractive attributes. It has a significant cargo carrying capacity. I think we're going to come onto that in a second in terms of its limitations, but most human genes can be made to fit. And it has a capsid which determines a lot of the tropism, exactly which cells it goes to, as well as its ability to evade a pre-existing immune response. And so there have been a number of exciting advances in capsid engineering, both using rational design and other techniques like directed evolution to evolve and engineer the capsid to modify its tropism. So in the eye, that might be the ability to be delivered, for example, intravitreally and overcome the inner limiting membrane and get to the retina, tropism more specifically towards key cell types such as photoreceptors. And there are also modifications that are known that can be made that reduce the likelihood that pre-existing antibodies will recognize that particular modified AAV. That's not to say that the AAV won't induce an immune response, but if you were to have already been exposed to some of the naturally occurring serotypes, it's possible to engineer the virus so that neutralization and reduced efficacy may not be observed. So the field has moved on very rapidly towards this. I think from a regulatory environment as well, the safety profile of AAV has proven to be extremely good. And so as we see these new vectors coming forwards, I think there'll be a positive regulatory environment that will result in their evaluation in clinic.
Great. The terms rational design and directed evolution are tossed around quite a bit in retinal gene therapy circles. Can you maybe explain and discuss some of the basic differences between these two strategies to produce second generation AAV?
Yes, certainly. So rational design really describes the process of understanding the biology and the molecular mechanisms by which AAV transduce tissues, able to show specific tropism. And then once those attributes are understood, you can deploy a rational approach to designing those into, for example, the capsid of AAV. So it's sort of taking existing knowledge, developing better understanding the biology of how AAV acts, and then assimilating that and designing rationally the capsid, for example, to overcome some of those issues. The best known rational design approach for AAV relates to the binding of AAV to heparan sulfate, which is found in the inner limiting membrane, would stop AAVs from getting from the vitreous, for example, into the actual retina itself. And so it's possible to rationally design in some mutations on the surface of the AAV capsid that stop it actually from binding heparan sulfate and therefore improve its penetration. In contrast, directed evolution is an approach where you create a vast number of variants, none of which necessarily need to be characterized, but you create a large pool of variants, and then you apply stringency to that in the way that evolution does, where you create a massive diversity and then apply stringency to that to find the best performers. So in this instance, you don't necessarily need to understand the biology. You use nature to select, if you like, the best variant with the attributes necessary to overcome the challenge that you put before it. And so it's a more agnostic approach in terms of understanding the biology, but nonetheless a very powerful technique that's been used outside the AAV field for many years, for example, in enzyme technology, to really define advances in the attributes that you want.
Well, thank you. Anybody else have anything to add?
Yeah, one of the other strategies, Peter mentioned very well, like the carrying capacity of AAV. Sometimes it could be limiting for some larger genes, but one of the other strategies that some people have come up with is actually splitting genes into multiple AAV. So you can actually divide it in half and rely on mechanisms like splicing or recombination to allow it to come back together. So that's another strategy to encompass larger genes.
I'm glad you brought that up, Glenn. So these split genes and mini gene approaches are interesting strategies in order to get around the cargo capacity of the AAV. So maybe you could elaborate just a bit on how would that work in reality? So you would actually inject into the eye a mixture of two vectors, each one carrying a different portion of the gene, and then you'd hope that cells would be transfected with both vectors, and then the genes would basically splice back together to form the full-length transgene. Can you take us through that? This might be just a bit more challenging than obviously a single vector with a small gene.
So I think from my standpoint, it is more challenging. It depends on the infective, like what the efficacy of your transduction vector is. So usually you will have a mixture, usually a 1-to-1 ratio. And then depending on what your efficacy is, if your efficacy is very high, then you have a high probability that both vector will get into every cell. But if your efficacy is very low, depending on what your vector is, that could be a challenge where some cells only got one of the two halves. But that is it depends on ultimately the goal, how many cells need to be transduced. If you only need 10% of your cells to be transduced to accomplish your goal, then the barrier is lower. But you are correct, Tom. It is difficult because you're mixing two different vector. Now you have two different products. I think the regulatory standpoint, that could be more complex. And then also, what is the correct ratio? It's not always a 1-to-1 optimally, depending on what the two halves look like. So there are definitely more challenges, but it is a way to get over that the size limitation that Peter mentioned.
Thanks. Peter mentioned a bit about rational design and directed evolution and how these techniques potentially could allow you to penetrate the internal limiting membrane and perhaps even minimize ocular inflammatory response. So Arshad and Jeff, you're both very busy clinicians and clinical investigators. You've been involved in lots of trials. You've been involved in lots of surgery and surgical innovation. As you know, historically, subretinal administration has been used for AAV gene therapies. Can you update us on some of the other delivery approaches that are evolving now?
Sure. It's as you noted, Tom, the approach to date had been one of two ways, either subretinal delivery via transvitreal approach, and there are a number of groups that have looked at that. Avalanche looked at it early on, and there were some issues with standardization of delivery of that, and they went on ultimately to intravitreal. And more recently, REGENXBIO has been looking at that for delivery of anti-VEGF therapy in neovascular AMD, and they've reported some very promising results from that. The other approach that has been frequently employed to date has been intravitreal delivery. And there have been several groups that have looked at that. Peter's group 4DMT is looking at it. Heier has looked at it. Adverum has looked at that as well. And again, there have been very promising results from that. But what some groups are looking at now is perhaps another approach, which is suprachoroidal delivery, either a needle delivery into the suprachoroidal space using Clearside's proprietary technology, or a subretinal delivery via suprachoroidal delivery, and that's via the Orbit device that was developed by J&J and is now part of the gyroscope technology, and Gyroscope is looking at that. Both Arshad and I are very heavily involved in all of those clinical trials that are looking at either suprachoroidal or subretinal via suprachoroidal delivery.
Well, thank you. Arshad, obviously you've had a lot of experience with some of these technologies. In your opinion, what are the potential risks and benefits of these various approaches, subretinal versus intravitreal versus suprachoroidal versus subretinal via a suprachoroidal approach? Any thoughts on that?
Yeah, absolutely. So I think Jeff summarized the delivery really well, and I'm lucky to be involved just like Jeff in all these programs. So obviously, subretinal delivery, REGENXBIO has shown really good data from their trials, and they're moving forward with that program. But obviously, the limitations are you have to go to the operating room and do a vitrectomy, and obviously that's not feasible for all of our patients with neovascular retinal diseases. So there's limitation in number of patients you can use that approach for. So the benefit is that we really haven't seen any serious adverse events or inflammation. There are some cases of retinal pigment changes, and we are still learning whether it's from the vector, whether it's from the bleb location. So there are some changes that are being made to address that. Going next to intravitreal delivery, as Peter and you discussed very nicely, the direct evolution was used for 7M8 vector by Adverum, ADVM022, to actually penetrate the ILM and really show robust protein production after intravitreal delivery. Obviously, that program has shown inflammation, especially in the high dose cohorts where, a third or so of patients have to be on chronic drops. And of course, initially in cohort 1 and 2, they used PO steroid prophylaxis and patients were having inflammation. So that's why topical drops were used in cohort 3 and 4. And we're learning more about it, but what Jeff and I have seen is excellent efficacy. And of course, the inflammation management is evolving. I think what we have learned is that we need to taper these patients off drops really, really slowly, because sometimes it can be an indolent kind of inflammation, not a frank uveitis. You can have, stable vision, but have some cells floating. So we are still learning about it, but I think we have data now from cohort 1, almost 92 weeks, and it has shown great efficacy with no rescue in these heavily pretreated population. Again, for the audience, the reminder is that in these trials, you're involving these hard to treat patients because this is early stage gene therapy trial, and both REGENXBIO as well as Adverum have shown robust efficacy. And Tom, you and Glenn have actually pioneered the suprachoroidal space for gene therapy. And I think your work has really advanced this space. And I think the question is that with the trials going on that Jeff and I are involved in, the AAVIATE and altitude trials in your neovascular AMD and DR, if suprachoroidal pans out where we see good efficacy and better safety than intravitreal, then it could be a potential compromise between surgery as well as intravitreal approach. So I think we're still learning. We don't have any data on suprachoroidal approaches at this point. So we'll have to wait and see, but really exciting times where gene therapy is actually now working, and this is a real thing. It's not a myth anymore. And we are seeing these patients who are chronically treated now going 1 or 2 years without getting any injections. So it's very exciting to be part of this really revolution of gene therapy.
If I could just add to Arshad's very eloquent description of these, it's the real world data on diseases like neovascular AMD and diabetic macular edema and venous occlusive disease as well has shown that undertreatment of these patients almost universally does not allow us to achieve the outcomes that we see in the phase 3 studies. And I think the reason we're all so excited about the potential of gene therapy, and there's still a lot of work to be done, is this is a means of dealing with the treatment burden and a means of achieving these long-term durabilities and long-term visual maintenance from the initial gains, and doing in a way where compliance is less of an issue. So I think all of us who treat these patients on a regular basis, Arshad, Glenn, Jose, myself, really understand the potential benefit of gene therapy in these diseases. And as we look at the potential of dry AMD treatments and recognizing those are patients who are going to need to be treated even longer, we recognize that gene therapy is really a potentially ideal approach for those diseases as well.
You brought up an excellent point. I'm actually just going to share a slide with everybody. It's research that I've just recently presented using the Vestrum Health Retina database. And it was 130,000 eyes that we looked at across 4 disease states. And you can see in the lower right quadrant is the cohort with neovascular AMD. And these are the real world outcomes at 1 year. So nearly 70,000 patients receiving 7.6 injections on average. And this is Vestrum Health, obviously, is a US-based retina specialist, and it gives us a real cross-section of what we're doing in the real world. And these patients are, as you can see, undertreated at 7.6 injections, and they only gain 3 letters. And across the spectrum, whether it's BRVO, CRVO, or DME, patients are generally undertreated, and the results fall far short of what we see in the randomized control trials that I'm displaying here. And then I'll just show one more slide. Again, I just presented this recently. And again, it's the same set of graphs. And these are, on the bottom right, you can see the 5-year cohort, the various cohorts out to 5 years for neovascular AMD. And what's really interesting is that on average, between 3 and 4 years, patients go negative. The average patient has lost vision in the real world by year 3.5. And again, you can see the same sort of trend with the other VEGF-mediated disorders, that they slowly lose vision. Your point is really well taken. We need better sustained release, and gene therapy may be a potential way to do that with the Biofactory. I think one of the most interesting innovations that I want to talk about briefly next is some of the work that Dr. Jose-Alain Sahel has done and really pioneered this. I think one of the challenges right now is when we do gene augmentation, that is replace genes for inherited retinal disease, we're approaching these disorders gene by gene. And then there are even some mutation-specific disorder innovations where we approach the disorders not only gene by gene, but mutation by mutation. And at some point, it becomes very difficult logistically to address these disorders, especially with over 260 genes involved. So I wanted Dr. Sahel to maybe comment on some of his really impressive work with a Biofactory approach, but not unlike Adverum and Regenix Bio, who are manufacturing anti-VEGF proteins locally in the retina, manufacturing a neurotrophic approach. In other words, to have a non-gene specific gene therapy to address a broader population of these patients with inherited retinal disease. So Jose, would you care to comment on some of this very important work?
Thank you, Tom. Well, the idea came from the fact that there are so many mutations, and although for the main mutation, we can hope that there would be a way to correct that if we take care of the patient early enough before the rods have undergone degeneration. There are also many patients that come to us at the stage where most of the rod photoreceptors have disappeared or have started to undergo a degenerative process, and it's not certain that a gene therapy would be able to target dying cells or certainly not dead cells. And so I started to focus many years ago on this idea that there is a common pathway in most patients with rod cone dystrophy, is that at some point the cones start to degenerate, and in many cases there is no gene defect underlying in the cone themselves. And so we found out after many years of research that actually there is a trophic factor that is made by rods and is supporting cone photoreceptors. We cloned this factor from the signaling, and it turns out that the loss of expression of this factor is happening anytime you lose more than 75% of rod outer segments. And so the idea is to bring back this factor so that you are not curing the disease. It's like the anti-VEGF therapy in AMD. You're not curing the disease. You are reacting at the amplifying stage of the disease, where the rods have disappeared mostly, and the cones are starting to degenerate. And so this is what we call rod-derived cone viability factor, that is now in preparation of a clinical trial, which is due to start at the end of this year, to try to protect the cones at the stage of the disease where you can no longer target the rods, mostly because you have too many gene defects or because the intervention might be too late. So in many animal models, from mice, rats, and pig, we found that this is protecting the structure and the function. The most important aspect is that it's maintaining the outer segment of cone photoreceptors. And as you know, this is where the phototransduction is occurring. Obviously, this is only animal data, and until we get the patient data, we won't know whether this is a valid approach or not in patients. But the advantage of that is that this is a biological factor that does exist in normal retina, that is disappearing when the rods are dying, and that potentially could rescue the cones. There is another gene-independent approach. Actually, I have to mention that other groups have been working on gene-independent approach. For example, Ocugen is working on an approach where they try to de-differentiate photoreceptors using NR2A3, which is an approach mostly trying to reverse the maturation of photoreceptors and trying to protect them. this could be a valid approach too. It's more or less differentiating the cells, so you wonder the functionality, but this looks kind of promising. There is also the approach that Pete Campocaro is using antioxidants to protect cone photoreceptors. It's not a gene therapy approach, but it could be of interest too. Once the cones have really undergone an advanced stage of degeneration, there is another gene-independent approach I've been working on, like others, which is optogenetics. Optogenetics, the goal is no longer to protect the cells, is to try to restore some function, either in the remaining cones, or if the cones have died, in the remaining cells in the retina, which could be the bipolar cells, or most often the ganglion cells. And we have an ongoing trial where we have shown safety actually with this intravitreal approach to deliver a vector to target ganglion cells. And hopefully we'll report soon on the efficacy on this approach, also in restoring some vision. So all of these are gene-independent approaches. As you pointed out, we are not curing the disease. We are just trying to stop the blindness, at least to reverse some of it, but just to avoid the loss of the central vision in these patients. So these are, I would say, complementary approaches. I wouldn't describe them as competing against the others, because clearly if you intervene early enough and you have a gene defect that is well identified, this is still the ideal cure for the patient, but in many cases, this may not be a feasible approach in patients.
Well, thank you, Jose. I find this optogenetics fascinating. These channel rhodopsins were originally, I guess, produced from or found in light-sensitive algae. So how would that work? I mean, obviously, you're producing a non-human, non-native protein. Would you expect inflammation with some of these approaches?
Yeah, you should expect, because this would be like synthetic biology. This doesn't exist in humans, so these are proteins that come from lower species. It turns out that clearly in the mouse there was no inflammation, in the rat there was none, in the primate there was very limited with mild immunosuppression, and now in the gene therapy trial that we have undergone, we are already at 7 patients, 3 dose escalation, and the inflammation is very mild. Actually, sometimes there is no inflammation at all, just with a steroid regimen as a classical one that you have in many gene therapy trials. Not very different from the gene therapy trial we did on Leber's optic neuropathy, for example. So this is a good surprise, actually. But there are 2 other issues with this approach. One is that these proteins require a lot of light and to be activated. And if you are using the green spectrum of light or the blue spectrum, the amount of light is almost toxic. So we shifted towards the red. We use the protein developed at MIT by Ted Boyden, and then you are 50 times lower than any safety, let's say, for ophthalmic examination. So this becomes a safe approach. The other limitation is that you need to replace all the amplification that occurs in natural phototransduction. As you know, 1 photon is leading to 1 million amplification, which you don't have with this system. And we are using goggles to send the light. Other groups like John Flannery are using other types of optogenes to try to not require this amplification of light. You still need the adaptation to changes in the levels of light, which is very important also in the retina. So this is a new area of investigation, but clearly a promising one for blind patients.
Thank you. The concept of inflammation that you just touched upon is a really important safety concern in retinal gene therapy. Not only potentially expressing foreign or non-native proteins that the immune system reacts against, but even the viral vectors themselves. And I always have to remind myself that, in some instances, we're injecting, hundreds of billions of viral vectors, of viral particles in the eye. So naturally, you'd expect some inflammation. What's really interesting and fortunate for retina is that the subretinal space has been considered immune privilege, which basically spurred on gene therapy research. So Glenn, you're an expert on inflammation, especially with respect to gene therapy. Can you elaborate a little bit on immune privilege, subretinal space versus suprachoroidal space versus intravitreal space, and maybe discuss some of the research that you've done?
Sure. Thanks, Tom. So generally, when we talk about inflammation, first of all, in the eye, we're primarily talking about clinical inflammation, signs of anterior chamber cell vitritis or chorioretinitis, although there's this entire aspect of inflammation that involves neutralizing antibodies, T cell responses that we don't clinically see, but we also know is occurring. Generally, ocular privilege, immune privilege refers to the fact that the eye is within a blood-retinal barrier formed by the tight junctions of the endothelial cells and retinal vessels, as well as the RPE. So anything that's delivered into the subretinal space is within that barrier, so it doesn't get exposed to immune cells. There's also aspects that are similar to what's known as ACADE, which is an anterior chamber immune deviation that allows corneal transplants to occur. That's why corneal transplants, the immune system in the eye is naturally immunosuppressive. So what we've known, at least prior to suprachoroidal, we know that the subretinal space is immune privilege, and the intravitreal injection is less so, because what happened is that a lot of the viral particles can exit the eye through the trabecular meshwork. And so it's actually, there's been studies that show that intravitreal injections could stimulate immune responses. And I'm talking about things like T cell response or neutralizing antibody responses that are similar to even like an intravenous injection. So it could be quite a lot of leakage of viral particles that leads to secondary immune inflammation like vitritis, et cetera, in the eye. Now, the suprachoroidal space is interesting because it's technically outside the blood-retinal barrier because it's outside the RPE. And one of the studies that we recently showed was that when we inject virus into that space, actually, it like subretinal injections, most of the viral particles do not go into circulation, which is good. So unlike intravitreal, which leaks out into the eye, it probably goes through the uveal scleral outflow. So most of it actually stays in the eye. So it doesn't cause as much inflammation or immune response to the AAV vector itself. But we do see that a lot of the transduction can occur in the sclera because the natural fluid is being pushed, from the inside of the eye out. So there are a lot of viral particles can be in the sclera, and you can have local transduction of the transgene. So what we found was that there was actually higher T cell response to the transgene that's being expressed in the sclera. And this is, in our study, we used GFP, which, as you alluded to, is a foreign protein. So it's not a great example of a measure of what kind of immune response you would get. So we did see a lot of inflammation, but we presume that if it were expressing like a human-based or a human protein or a protein that normally is expressed in humans, we might not expect that to occur. So it's a very unique compartment that has some of the advantages of subretinal space, but it also has some of the disadvantage of being expressed outside the blood-retinal barrier.
Tom, can I ask Glenn a couple questions? Because nobody knows this area better than Glenn. And inflammation is obviously such a critical component of the success of gene therapy. Glenn, I've noticed inflammation seems to come in a couple of different patterns. There's sometimes when we inject and you see inflammation within the first week. And then there's, especially in intravitreal and perhaps in some of the other techniques as well, I've seen inflammation at about a month after. And I've always taken that inflammation at a month after to be a sign of expression. And I'm interested in your thoughts on this.
That's actually a really good question. To be really honest, I think there's a lot of discrepancy in the literature, and it's hard to say just based on the timing or whatever that the inflammation corresponds to what system, like at the molecular level, what's triggering that inflammation. Because again, as I mentioned, the clinical and the molecular effects might not be so well, it's not so clear in every patient. But I do believe that if you see inflammation very rapidly within a week, it's unlikely to be transgene expression just because it takes time for the transgenes to be made. So we might expect that to be more a result of the viral particle itself. Whereas presumably the transgene, usually AAV, start making it at 2 to 3 weeks, and at 1 month, it's where it usually peaks. So if you see inflammation at 1 month out, it could be more likely related to the transgene. But I don't think you know for sure. And I think those studies are ongoing, but it's a very murky field, inflammation in viral gene therapy.
Can I add a comment? here as well, and very nicely described, Glenn. I think some of the other things that we're seeing as advances are improvements in manufacturing. Some important points here. I mean, this is a very complex biologic. You've got capsid, you've got transgene, you've got all the excipients and so on that go along with the product. And what we're seeing, I think, with more modern techniques for manufacturing is not only a reduction and improved impurities, which may themselves be pro-inflammatory, so we're seeing a reduction there. But we have an unusual phenomenon in the manufacturing process of gene therapy where you get empty capsids. So you end up producing a lot of capsids which are themselves whole, but in some cases they're not packaged with the transgene. So effectively you end up delivering a lot of additional protein which could be inflammatory, and not so much of your actual transgene, which will be which has therapeutic potential. But I think with modern manufacturing techniques, we're now getting to a very, very high ratio of capsids containing the therapeutic, and I think that should help to reduce inflammatory responses moving forwards.
That's a good point. So at Spark for Luxturna, the empty capsids were removed, and that was thought to be not only helpful for inflammation potentially, but also to enhance efficacy in case the binding sites on the RPE cells were limiting. You didn't want to occupy those binding sites with empty capsids. But that's a really good point. So while we're touching on inflammation, Peter, do you want to go back to where we started and maybe talk a little bit about how directed evolution might minimize inflammation with intravitreal route?
Sure. I think it's a really interesting question, and I think Glenn and Jeff have described well the complexities of what we mean by inflammation. But if we assume that in part this has an immune aspect to it, then going back to what I was describing earlier, which are essentially surface residues on the capsid, which act as epitopes and are potentially immunogenic, it is possible to use directed evolution to identify capsids which are devoid of those epitopes that might previously have been exposed to the individual, and therefore wouldn't excite an immune response by virtue of being previously sensitized to that capsid. Beyond that, there is the possibility with directed evolution and rational design that by improving targeting to key cell types, you minimize your off-target transduction. By which I mean, if we were able to target much more effectively and selectively photoreceptor or rod photoreceptor transduction, thereby minimizing the amount of vector that would get to potentially antigen-presenting cells like the RPE or so on, you could mitigate against some of those inflammatory responses. So I think there's sort of a multi-pronged approach to the potential to reduce these inflammatory and potentially immune responses with modern therapeutic development.
Can I ask a question to Peter and Glenn? There've been talk, and you also, because you were involved with subretinal treatment also. My question is that there is a talk about, so as Glenn said, clinical signs of inflammation versus molecular signs of inflammation. So when you are delivering, billions of vector in a localized subretinal space, obviously we are not seeing clinical inflammation. Has anybody looked at, in terms of molecular level, what's really going in that localized area? Do we have subclinical or molecular inflammation that we are not seeing? And the reason I'm asking is because if you're having pigment changes, my interest is what's causing that. Is it burnout of the cell? Is it inflammatory? And, I thought, since you are both experts in inflammation, if you have any thoughts on that.
Yeah, so we definitely see, for example, with some of our studies, we've seen actually like kind of focal chorioretinitis, like little yellowish spots, which in monkeys, maybe in humans will be more of these kind of pigment changes you're referring to. They are actually little focal areas of inflammation, and that could be inflammatory cells infiltrating. But again, then we start looking at, like, is it the T cell response or is it a, B cell? The more I learn about it, the more questions I have. I do feel like, all of us were just kind of still just grasping at the beginning of it. I feel like we're just at the surface of understanding that there are both vector as well as transgene elements that are triggering the inflammation, but how that leads to specifically the cellular infiltration. I do agree, even if you don't see clinical signs of inflammation or robust amounts of vitritis or something, there could be a small amount of inflammatory infiltrate that we're not seeing. The question is, does it really affect the outcome? As long as, if we treat it, it might not, there's been studies showing that neutralizing antibodies might not have much effect on the actual clinical outcome. So it might not be a big deal ultimately, but it is important for us to understand it more.
I think there's also, when you think about how much inflammation you'll tolerate, a lot has to do with gene therapy versus, say, intravitreal injections. If I see several weeks or even 4 to 6 weeks of inflammation associated with an injection or something that even lasts 3 to 6 months, that might not be so tolerable. But if we're seeing 6 weeks of inflammation that can be controlled either with orals or topicals, or even, safer immunosuppressives, and that leads to a year, 2 years, 3 years of efficacy, I think that would be something that many clinicians and many patients would well tolerate. And as Arshad said, we're seeing efficacy certainly in the wet AMD studies with REGENXBIO and Adverum of patients well past a year, and some of the REGENX patients out 2+ years with strong anatomic benefits and efficacy. So as long as we can control the inflammation, I think it's tolerable.
I wanted to shift gears a little bit because we only have a few minutes remaining. We've talked a lot about gene augmentation, but I want to talk a little bit about some of the other strategies, including gene editing, gene inactivation, silencing a gene, often a dominant negative one, and even touch upon RNA therapeutics. Obviously, we can't do these topics justice with the remaining time, but maybe just we could just discuss briefly about gene editing, what it is, and there are actually some trials now employing the strategies. It's really fascinating. Glenn, I think you've done some work in this area. Do you have any comments to start?
Yeah, I think our current first generation of gene therapies are mostly, as you mentioned, gene augmentation. So there's a missing gene or a broken gene, and we're giving a working copy back. But there are a lot of diseases, as you mentioned, these dominant diseases where the gene is there, it's just that it's doing something bad and you want to actually destroy it. So I like the concept of gene editing because gene editing involves an enzyme that's derived from bacteria that actually will manipulate and can ablate a gene. And our lab's been using it to try to target VEGF, but people like Steve Zhang from Columbia actually has this concept of for these dominant diseases where there are different mutations, you can just ablate the gene, independent of whether, what mutation it is, and then replace it. We call it the ablate and replace approach. I think that gene editing could be potentially a way kind of similar to what Jose was mentioning, kind of like a more mutation agnostic approach to treat a variety of diseases. So that's definitely some of the things that are on the horizon. Editas, I think, was what you're talking about with the CEP290. That's one of the first kind of a dominant, I think it's a splice mutation that they're trying to cut out with that gene editing technique. But I think that's the only one that I know of currently in eyes in human trials. And I think there's going to be a lot more optimization going forward.
Anyone else care to elaborate either on gene editing or knock down and replace strategy with the short hairpin RNA? On
the gene editing approach, just to build, that's a very nice description from Glenn. I think the challenges we have right at the moment, the ideal would be to actually be able to correct the genetic mutation. There is technology that can do that, at least in vitro. The main challenges remain the efficiency at which you can achieve this. And actually, going back to a very earlier point you talked about, the size of the AAV genome. It's not possible to package a cut and correct gene editing approach right now in a single vector. So you're moving into that sort of that dual vector range, and some types of mutations are not readily correctable. So it's a fantastically exciting technology, but it's certainly significantly limited at the current time.
I think you're right. I mean, well, there is probably another indication that is upcoming, which is H2A, where also the same approach of gene editing is upcoming and quite promising. I think Arlewin has been developing an approach to correct some rhodopsin mutations using similar technologies, not exactly identical. What is promising and kind of surprising when I think it was 2 years ago in Nature Medicine, there was both the CEP290 and the gene editing approach by Editas that was published. So I wrote an editorial on that. The idea of using RNA technologies, the Procure approach, for example, is also very promising. Interestingly, the effect is lasting much longer than what was predicted. And one of the positive aspects of that is that it looks like these ciliopathies, which many people thought were irreversible, like developmental defects, there is the likelihood that you could regrow some of your outer segments using these technologies and reverse some of the phenotype of the disease. So these are very targeted approaches in contrast to what I was describing, which is more generic, but the possibilities to correct the gene defect and to regrow, at least in part, the connecting cilium and some of the outer segment is kind of there. So these RNA technologies are competing in some ways with gene editing, quite elegant, as we can see, and that could be designed for specific gene defects. I think the main issue we're going to face is that if you are getting into mutation-specific approaches, the landscape is going to become even more complex. How many diseases can you tackle with that, especially when you're talking about ultra-rare conditions with very few patients? And this is where, hopefully in the future, the development of vectors that will appear are very safe, cell-specific promoters that are going to be cell-specific might make the pathway to developing these gene therapies for only a few patients feasible. If not, the cost is going to be prohibitive, and these patients will become orphaned inside the field of orphan disease. So we need to find a way to make this affordable, and which means that developing vectors that are going to be recognized as safe for rod photoreceptors, cone photoreceptors, whatever cell type, and being able to make sure that we can control the titer, the inflammation, the efficiency, hopefully will pave a way for many gene defects to be targeted with some sort of generic approach. Besides what I was describing of a gene-independent approach, which also is complementing this landscape.
Thank you, Jose. Well, I guess for our last question, I'm going to ask each of the panelists to give us their prediction for retinal gene therapy 10 years from now. And maybe I'll just go down alphabetically. Peter, any thoughts about where you see retina gene therapy heading, perhaps over the next decade?
Well, thank you, Tom. Yes, I think there's an exciting future for gene therapy. I think gene therapy has gone through the same kind of process we've seen across medicine, where there are some initial successes and then the field struggles to really make it sort of mainline, and then those technological issues are overcome, and we see multiple therapies growing out from this. And I think we can look forward to that in ocular and retinal gene therapy. I think from the pharmaceutical end of things, there's always a tremendous attraction about the ocular space. From a manufacturing and a commercial perspective, you need small volumes, small manufacturing hurdles to be overcome, in contrast to, for example, intravenous gene therapy approaches. So there's a lot of attraction there. So I think we're going to see continued interest, continued growth. I think we're likely to see multiple additional approvals. For me, I think it's the discovery that gene therapy could really be beneficial for some of the large complex diseases like wet macular degeneration, dry AMD. I think those could be really transformative for the retinal field and have grown out of the sort of single gene approaches we've seen. And I think in the next 10 years, that's probably going to be the most exciting area for retinal gene therapy.
Jeff Heier?
Sure. I think I share everybody's enthusiasm. I think that we're starting to see successes in gene therapy that are not for small percentages of patients, but are for the large percentage of patients. You know, in the higher dose cohorts in REGENXBIO, we're seeing the majority of patients respond. Similar with Adverum, we're seeing the majority of patients respond. So I think there's a very good chance that we're going to see treatments for wet AMD and subsequently DME and perhaps DR and RVO that allow us to treat significant portions of patients with these treatments. I think the natural progression will be to dry AMD, and I think we're probably on the verge of seeing successful treatments for dry AMD, where the complement inhibitors look like they're promising. And I think the next step will be looking at gene therapy to tackle those diseases. So I think in 10 years, we're likely to see a fair number of patients undergoing gene therapy for treatment of their disease.
Arshad, do you have anything to add?
No, I think I agree with what Jeff and Peter have mentioned. I think based on the efficacy and durability we are seeing currently and a manageable safety profile of these different programs, I think even before 10 years, Tom, we may have an FDA-approved treatment for wet AMD and likely DME and DR. I think, DR is really exciting, right? Because it's a lifetime disease and patients tend to be younger. And I think if we can have a delivery approach that is easy and we have good efficacy and manageable safety of an approach, I think it's going to be really exciting. So I, just like Jeff, feel like majority of our patients will be getting gene therapy 5 to 10 years from now. One thing Jeff mentioned, obviously, is dry AMD. And, a lot of our patients with wet AMD end up with geographic atrophy down the road. And that's a concurrent progression, in my opinion, of dry AMD progression versus the anti-VEGF effect. And I think if you have a dry AMD gene therapy as well as you have wet AMD gene therapy, and what Glenn and Peter were talking earlier, or you, it's like, can we give both of those treatments to a patient, and can we maintain good visual acuity in these patients who are treated for their wet? So I think it's really exciting time. I think the delivery approaches as well as AAV vectors are going to continue to improve. And I think if we can address inflammation, and as Jeff said, we have not seen severe inflammation, no vasculitis in the Adverum program. And I think if we can learn how to manage these patients well, I think it is going to be a clear win for patients and really game changing for our fields, where we can really help preserve vision long-term for these patients.
No, I agree. It's very exciting to think that we can have a gene therapy function as sustained release for some of these large multifactorial disorders. Jose, as I mentioned earlier, I'm particularly excited about some of these gene-independent approaches. Where do you see that 10 years from now in order to improve the logistics of treating so many of these genetic defects? How do you see that panning out with the various gene-independent approaches you've discussed over the next 10 years?
Well, I think clearly it will complement the pipeline, and we would be able to treat at many stages of the disease versus targeting only some windows of opportunity. So this would be a very important aspect. If we want to tackle large disease, I don't want to repeat what has been said, but we have also to consider the possibility of combination therapies and of tunable therapies, because at some point you may want to turn off a gene that you are administering, especially for wet AMD as an example, but this may be true. Gene regulation is going to be important, and this will require vectors that could accommodate larger genomes to be able to really incorporate into the same vector, maybe a combination of therapy, corrective GNP, neuroprotection therapy, immunomodulation. You could think about many things you could put into the same vector, but at the same time, you would need to be able to regulate that. And so it's going to be more complex, but also more personalized in terms of stage of the disease and specific need of this individual patient.
Thank you. And Glenn, anything to add to the great comments we've gotten so far?
Yeah, not much to add, but I do want to echo Jose's comment. I think we're in the first inning. I think we're going to go for precision. I think things like CRISPR, you only need to make one edit and you don't need it to be expressed forever. So it could be, the idea of tuning, time, something you can turn on and off, something you can target to specific cells as opposed to every cell possible. I think those are all areas of research that'll be making this more interesting in the next 10 years.
Well, thank you very much. I want to thank the panel, Really, what a wonderful panel of world-class experts to discuss an extremely exciting time in retina and in gene therapy innovation. Thank you so much for your very insightful comments.
Thank you.
Thank you, Tom.
Thank you. Thanks, Tom.
Thanks for listening, and thank you to our host, Dr. Tom Chula, and of course, our thanks to the panel, Drs. Francis, Heier, Khanani, Sahel, and Yiu for sharing their valuable insight. It's exciting to think about the impact gene therapy will have on patients' lives. We can't wait to see what the future holds. If you enjoyed this episode, please subscribe, rate, and share us with your colleagues to help spread the word. And if you'd like to be a guest on our show, go to OIS.net and click Get Involved. Until next week, keep innovating.